So for years, we've been using animal models to study human diseases, and obviously a mouse is not a human. So in this particular endeavor, we're starting to work towards building better models using human cells to study human diseases.
There's a real opportunity now to capitalise on the advancement that's happening in the technology we're working on, which is organoids, which are essentially mini replicating organs in a dish. So they're self-replicating and they can recapitulate all of the cell types and the architecture in a human, say, lung or gut, for example. Why that's really important is so that we can model infections in particular that are pathogens that will only infect human cells and you could never have an animal model for.
One of the key things that we're really working towards is standardizing protocols and making sure that people are doing the same thing across jurisdictions. We started an initiative within Canada called the Canadian National Organoid Network with this in mind. And when this grant opportunity came about, we realized that this was an opportunity to extend that influence in terms of standardization and developing new protocols for investigators to use.
So one of the key outcomes from this project is standardising the way that our labs do these organoid experiments. But that needs to be at the level of the people who are doing the experiments at the bench.
We're working towards continual exchanges, having students come across and train in each other's labs, and I think that's going to lead to some long-term collaborations and some really good model development and knowledge exchange throughout. And the hope is what we're doing here can actually set the stage for something bigger in terms of networks for sharing of reagents, sharing of biospecimens to really coordinate efforts for the next pandemic or the next big health emergency.